Hello and welcome back. In this section, we are going to learn the detailed physiology of vision, okay, which is not this one slide. This one slide is like high school physiology of vision. I don't know, maybe not even that. It's just like Sparks notes, but you need to be able to put in all the little details like chemical reactions, what chemicals are we talking about, what transmitters are we talking about, what's happening everywhere. So let's get into that. Let's talk about those chemical reactions. But before we get into all those chemical reactions, we need to talk first about those photopigments that are going to absorb the light and what we mean by light energy to nerve impulses. So the photopigments, to refresh your memory, these were specific molecules that are going to be absorbing the light energy, and they're in that outer plasma membrane. On that outer membrane. So on the rods and cones, the outer segment, right up in there in the plasma membrane, that's where all those photopigments are located. So in rods, the name of that photopigment is rhodopsin. So R for rods, rhodopsin, okay? And rhodopsin is this black dotted line right here representing the rods. So it's gonna be most sensitive to light around the 500 to 550 nanometer wavelength. Don't have to memorize the numbers. I just want you to realize it's sort of right in the middle. Now, as opposed to rhodopsin in rods, in cones, we have a different photopigment called photopsin. And there actually are 3 different photopsin proteins. Of course, each one has a different name. We're not going to learn the names, so just learn them all as a photopsin. But each of those for the 3 basic cones absorb at a different wavelength. And by the way, there's like 10% of the population that has more than 3 basic colors. They have 4 basic colors, which is really kind of cool, but let's stick with the basic 3 colors. So we have a lower wavelength being absorbed by blue cones, slightly higher than the rods by the green cones, and even higher still by the red cones. Okay. So if you go from the right, which has the highest, to the lowest being on the left, basically that's your Roy G. Biv that you learned about when you learned the colors of the rainbow, ROYGBIV, red, orange, yellow, green, blue, indigo, violet, Roy G. Biv. Okay. So this doesn't mean that only that color cone can do that one, but like, do you see how the green and red cones overlap? Okay, so the green cones are better at a slightly lower wavelength than the red cones are, but there is some overlap in them. Okay, so how do we perceive all these colors in between them? Well, basically we're getting nerve stimulation from a bunch of different ones in different ratios. Okay, there's no one type of cone that's maximally stimulated. Very rare circumstances are only the blues are going to be stimulated, and your brain's going to interpret it. Okay. So that brings us to the concept called phototransduction. And what this means is we're changing energy forms. We're going from light energy to an electrical signal, which gives us a nerve impulse. Okay. So in order to understand how this happens, okay, we're not going to do a bunch of physics. There's physics involved. We're going to skip the physics. Okay. We're going to talk about what's happening in the photopigment when it's exposed to light. So we're still not going through the sequence of equations. We just need to kind of look at the photopigment and see what's happening to its conformation. So this is called, the reaction we want to talk about is called a bleaching reaction, and happens in rods. So before you turn the lights on, so like when you're looking far away in dim light, the rhodopsin is in this shape up there at 12 o'clock called cis-retinol. And it has this slightly bent shape. And it has this slightly bent shape because on the carbon chain, certain molecules are all hanging on one side. Okay. Now, when it is hit by the light waves, when it hits those photoreceptors, the retinol changes shape. It changes from a cis shape, which means stuff is all the same, to a trans shape, which is going to give us a more linear retinol, which is in the box at 3 o'clock. And if you look at the box at 3 o'clock, what you will notice, every other carbon has its group on the opposite side. So they're always opposite each other, thus giving us the trans shape. So you need to compare the trans shape versus the cis side. You'll understand on the cis, we've got places where we have attachments on the same side, and that gives us that curvature. Okay, so when we have switched from, when light has made us move from a cis to a trans retinol, basically what is going to happen is that trans retinol breaks off of the opsin. Okay? And that's where transduction happens. This is what we're calling the bleaching reaction. Okay. So the dissociation of the rhodopsin, so that first compound, the rhodopsin found in rods, it's hit by light. The cis retinol becomes a trans retinol, and so instead of having one rhodopsin, now we have two products. We have a trans retinol and we have an opsin. And in doing that, this rhodopsin changes color from this lovely bluish purple color to becoming almost clearless. And that's why it's called the bleaching reaction, because it just changes colors. Like bleach takes the color out of anything. Okay. And so what this does, when the light hits that, because the retinol has changed from cis to trans, and thereby the retinol has separated from the rhodopsin, we don't have as much rhodopsin. So now the rods have a much lower level of rhodopsin in that, and so that affects our ability to see in the dim, because we just don't have the rhodopsin. If you don't have rhodopsin in the rods, then the rods can't do what they're supposed to do. And so in order for the rods to see vision, then we have to figure out how can we get rhodopsin made again? How can we get more retinol to attach to opsin to give us the rhodopsin again? Okay. And so for the regeneration, what happens is you go into low light or dark light, and we use our neighborhood energy source, ATP, and that will change the trans retinol back to cis retinol, and it's all bent up, and so it can rebind with an opsin to reform a rhodopsin. So light breaks it apart, it changes the shape, breaks it apart. ATP will change the shape back, and it can come back together. Okay, and it sounds like it'd be easy, go from A to B, then B to A, A to B, B to A. However, it's A to B, and B to A. It's a very slow process to go back, to regenerate it, okay? After 5 minutes, only 50% of it is regenerated, okay? Now, the same process happens in rods, only of course it's not called rhodopsin, it's called rhodopsin. And so I'm not gonna go through it, but it has the same sort of process. However, in cones, the regeneration is super quick. So that's why cones are great for bright light. Okay, so this fact that we can't see well in dim light, this is called dark adaptation, the slow return of sensitivity going from bright light to dimness, okay? So when we look at cones and compare them to rods, okay? Initially, what you're gonna see is you're gonna see the cones are becoming less functional because they need brighter light to stimulate them, okay? So if you have bright lights on at time 0, if you're sitting in a room with bright lights at time 0, then you turn off the lights, okay? What's happening is you're losing your cone. The first 7 minutes, basically you are losing cones, harder to see. Rods are still bleached. They're starting to get back, they're starting to get back, they're starting to get back a little bit, but it's not until about the 7-minute mark where the rods are then gonna start taking over the vision, okay? And it really takes 20 minutes to totally see well in the dark, okay? So what does that mean? Don't drive around with your lights off, okay? It'll take you 20 minutes before your eyeballs can adjust to the low light levels. Just don't even try it. It also means that totally explains why when you walk into a dark theater and looking for your friends, they can be up there waving, hey, we're over here, and you're just standing there and going, I can't see a thing, I can't see a thing, I can't see a thing, because I don't have any rhodopsin in my rods. Okay, so that's the dark adaptation. Now, light adaptation would then be the opposite. You've been sitting in the dark, watching the dark movie, and all of a sudden, the lights come on, okay? So your eyes have to adjust from low light to bright light. So the first thing that's gonna happen, your pupils are gonna go, okay, to cut down the amount of light entering them. But you still can have a temporary blindness. I think you all know that when somebody takes a flashlight and they put it in your eyeballs, go, wait a second, and then you can't see anything, okay? So what is happening is, as soon as there is that light, those rods are going to become inactivated. And initially the cones are oversensitive, and then they'll gradually readjust to the light. Okay, so that's all that's showing. Okay, so after about 5 to 10 minutes, the cones, remember, they're faster. Okay, so it took us a full 20 minutes for the rods to fully adjust to the dark light, although it was like 7 minutes to kind of get things going. After 5 minutes, the cones are pretty good. After 10 minutes, like, they are fantastic. Okay, now we're ready to chart some chemical reactions. So if you need to go back and review that, go back and review that. So this, as you can see, is a rod. So let's talk about how this rod is going to do phototransduction in the dark. How this rod in the dark is going to take some light energy and make you be able to see. Okay. So remember, everything starts way out here in the outer segment. Now, out here in the outer segment of the rod, it's constantly taking it's constantly making cyclic GMP. So it's using GTP, not ATP, but GTP is an energy source, and it's making cyclic GMP from it and storing there. So we have really high levels of cyclic GMP in this outer segment of the rod. And that cyclic GMP is going to bind to cation channels. Okay? And when it binds to a cation channel, that opens it. And that allows both calcium and sodium to enter. Okay, now when you look at a lot of pictures online, they're just going to show sodium entering, but I want you to realize both sodium and calcium can enter through that open cation channel. Okay, and so when sodium and calcium enter, this is called the dark current because it's happening in the dark. Okay, so the entry of all those cations depolarizes the photoreceptor up to minus 40 V. Millivolts. Okay, so from where it's really polarized, now it's been depolarized to minus 40, and that's enough to start a graded potential, a local potential, which will then reach voltage gated calcium channels located way down at the other end of the rod. So that graded potential goes from the outer segment to the inner segment through the cell body, down where the axon, all the way down to where the axon terminals and those voltage gated calcium channels are located. Okay, so when that potential reaches there, that's going to cause the calcium channels to open. So now calcium is entering the terminal bouton area. Okay, and that causes little synaptic vesicles filled with neurotransmitter to release glutamate, the neurotransmitter involved. So the glutamate's released into the synaptic cleft, looking for its receptors on the postsynaptic neuron, which in this case is a bipolar cell. So the glutamate is going to bind to receptors on the bipolar cell. Okay, so that was our first action potential. Now we're in our primary neuron. Okay, so when it binds, when glutamate binds to the bipolar cell, that causes hyperpolarization, not depolarization, sorry, hyperpolarization going down. Ooh, more negative. Okay. So it's inhibiting the bipolar cell. Thanks. Okay. And so because that bipolar cell is inhibited, it cannot release any neurotransmitters at its synapse with the ganglion cell. Therefore, there's no nerve signal given by the ganglion cells. So vision in the dark is by blocking their neurotransmitters. Kind of a novel concept, isn't it? All right, now in the light, totally different process going on. Okay, so make sure you understand that one before you move to the next one. We've got the same rod connected to a bipolar cell, connected to a ganglion cell. So we still have lots of cyclic GMP in there, okay? But now we've been exposed to the light. And so we have that bleaching phenomenon. And that bleaching phenomenon leads us to a drop in cyclic GMP, which closes cation channels. Okay. So how is this going to happen? This involves a G protein. So with that bleaching reaction, we're splitting the rhodopsin, okay? So we've gone from the rhodopsin with the cis retinol to now we have the trans retinol and the opsin over there. Okay. That activates a G protein. So you know we're in a second messenger system, okay? But it doesn't have anything to do with cyclic AMP. It doesn't have anything to do with adenylate cyclase. What it's going to activate is the enzyme phosphodiesterase, okay? Because G proteins activate enzymes. In this case, phosphodiesterase, which should be a PDE someplace over here. Yes, there's your PDE, okay? So phosphodiesterase breaks down cyclic GMP to GMP. So that's how we're getting our drop in cyclic GMP. Okay, so the bleaching activates the G protein, which causes cyclic GMP to fall off. Okay. The lower levels of cyclic GMP close those cation channels we talked about. No sodium coming in, no calcium coming in. Everything staying on the other side. No dark current. Okay. So now the photoreceptor is hyperpolarizing. So when it's hyperpolarized, that voltage channel way down by the axon terminal, it's closed. There's nothing come in. There's no synaptic vesicle releasing any neurotransmitter. So there's no glutamate leaving the rod and cone to activate the bipolar cell. That's not happening. Okay. So if that's not happening, that means the bipolar cell still gets to do its stuff. They're not being inhibited by the glutamate. So the bipolar cells themselves can release their neurotransmitter, which is also glutamate, and this glutamate binds to receptors on the ganglion cell, and if it reaches its threshold, here the glutamate's excitatory. And so if that ganglion cell reaches its threshold, it's going to propagate an impulse which travels along the axon from the secondary neuron to the tertiary neuron to its final destination in the brain. Okay. So here's two pictures, the dark on the left, the light on the right, left off all the other words. Once you understand the process, sit here. There's a lot of hints obviously here, and there's also voltage mapping going on here to help you remember what's going on. And this is just showing the rod and cone. So make sure you can carry it on to the bipolar cell and to the ganglion cell, and be sure to add in not only sodium, which they're showing, but also calcium, where calcium is entering everywhere on both processes. So at this point, that's the end of this section. So go ahead and stop it and review it. And I will see you again shortly. And thank you once again for all your hard work, because I know this section will take you a little bit of time to get it.